Okay, and then Kate, I'm gonna change that thing again so you don't have to see people join.
Yeah. This new
No, that was on Monday. Or no, that was on Monday.
No, it's not that thing. It's
Yeah.
in here. Yeah, can you hear and see? I just turned the camera on. Do you think that looks good? Just go up now and say something in there. Go for it. You're going to be broadcast on YouTube forever.
Yeah.
Yeah, but I can see it. Okay, here, I'll go up and you can view for everyone else. Closer to the mic. Oh shit, I don't know how What would you have it coming through?
Can you turn your speakers on?
Oh no, not that. Oh, okay. Brian, will you send me a text message so I can make sure my computer isn't broadcasting the noise? Will you send me a text? Great, it works. No, it's not.
I know.
Hi, Noah. Bye, Noah. Cool.
We're running out of juice. Oh, yeah, sure.
Running around.
I'm super
So she'll make a presentation, public questions, and we talk through thriller, make sure it's in the right place.
Oh, yeah, I was like, why are you making this? You can add it to
Like that farm.
Yeah.
Good afternoon. Hi there, everybody. Thank you for making it. My name is Stuart Sandin, and I've got the distinct honor of introducing you to the main event today, which I'll introduce in a moment. You know that we're here for the doctoral defense of Catherine Furby. She's going to be presenting, giving a presentation. But beforehand, I want to just give a little introduction to her. And by saying introduction to her, I want to introduce you to a person named Dr. Nalini Nadkarni. Odd as that. Nalini Nadkarni is a pretty famous forest ecologist. And I was first, she was a professor at Evergreen College. She's been really prominent forest ecologist in terms of sharing stories about the forest world, the rainforest, the rainforests of Washington State, etc. Her accomplishments include things like giving TED Talks and introducing non-traditional audiences to science and ecology, going into prisons and using forest ecology as a way to captivate inmates and to try to give some continuing education. She's had videos. She's even, some of her research was even attracted the attention of Ben Affleck, who was in one of these PBS shows that she did, PBS series that she had. But it's not really her final her accomplishments over her whole career that I want to tell you about. I want to go back in time to when I was a student, and my father took a copy of Discover magazine and pushed it over to me and said, "Hey, you have to read about this person, Dr. Nadkarni. She's a fascinating individual," and I learned something more about her. She was a young faculty at that point, and her story was different than most. She said, "I was an undergraduate and kind of came and went from school. I was a dancer, professional dancer. I went to school, I loved biology, and I'm here right now, and I'm at a crossroads," she said for herself. She said, "What am I going to do? I could go on to college, and I could be a biologist, or I could become a professional dancer." And if you look at it in sort of like a either-or standpoint, you know, she took the or. She was a professor of biology. But she merged the two. She felt that she really took her discipline, and she took biology and tried to bring her ability to dance, motion, etc., to the field that she worked in. Her specialty was epiphytic plants, the plants that live up in the canopy of rainforests. And for those of you that have been to a rainforest know that that's not right there. It's really high up. They're really tall trees. She used rope climbing or rock climbing equipment to levitate herself up to the canopy, and she would glide from tree to tree, from branch to branch, using her dancing skills to learn about these plants. She got 2 plants named after her that are from that because of her illustrious career studying these elephant plants. My dad introduced me to this probably as a way to investigate myself, to say, like, what's my silly son doing in this field that's called ecology? Why doesn't he do a real profession like going engineering or something like that? But he also was just fascinated by her story, her ability to merge 2 disciplines. And really, it is a captivating story, one that led to a ton of success. Seems like an aside, but then enters conversations that I have with Kate Furby. She comes to me and says, hey, I'm thinking about a story. I'm thinking about going into graduate school in biology, but I'm not convinced. I was an art major and a biology major at Bennington College. I've worked in aquaria, doing interpretive stuff and the biological side of things, and then traveled the globe thinking about these issues. I don't know what to do. Come on, come to graduate school, you can do it. And we all made that stupid decision, so come on. And she comes over here, and she completes what is a terrific thesis, and you'll hear more about it, looking at a coral reef, but not from the standpoint that we see most people treating it as superficial view, both literally and figuratively, but instead an involved view, getting into the reef, getting inside the coral, studying who are the organisms that are making the play go. You'll see more of this. But she never abandoned, she never let the science get her away from art. And you'll see that ubiquitously throughout her thesis, or throughout this presentation. But during her PhD time, she didn't let the artistic view, she didn't let her fascination with the beauty of nature and the communication of it, and really the artistry that exists in nature and the communication of it, she never let that go. She participated in a lot of science education training programs, multiple places. I think it was for a while I thought it was a scam, but then she got, because she was traveling, like, oh, you're in Montana, okay. But then she ends up teaching classes, getting invited places, getting paid to teach on ways to creatively communicate science to a broader audience. She creates videos, YouTube, she's a YouTube sensation, you know? She's got a channel, CK Run. And for those of you who know it, you should look her up and be one of the 100,000 people that have done it. And for the rest of you that aren't millennials, that's something having to do with the internet that I know nothing about. So move on. She's given invited presentation. She as well was presented at a TEDx symposium, gave a stunning talk. Was able to share her passion and her story with a broad audience. And she's rubbed elbows with amongst the most famous people in the Explorers Club in New York City, you know, representing Scripps Institution of Oceanography and telling stories, getting the science across. I'm really proud of Kate. She's been a wonder to have in the lab. It's been terrific to have both the scientific insight, curiosity, and passion in the lab, but then also that groundedness, that ability to take science and to share it, and to help us to share things, to get us out of the ivory tower. And it's going to be something where we'll remember her and we'll appreciate it, and it'll be sad when she's gone from the lab. We'll have her at least for another month or so. So she does plenty of great work, and that's what we're going to see today. The one thing that probably is going to be the biggest memory that she's going to leave in the lab is this inordinate passion for the coral called fungia. Okay, this has nothing to do with being a millennial right now. Probably millennial is a bad part in this whole thing. But if she did teach us all to love fungia, I'm going to use that as a teaser. If you need something to talk to her about at the party that will follow this event, right, then that's something to ask her about. Fungia, right down. It sounds like fun. The format that we have right now, Kate is going to give a public presentation of her thesis. It's an overview of the work that she completed that is a highlight reel. We're going to keep her committee, who I thank for being here, multidisciplinary, multi-department committee, representing Kate's multidisciplinary, multi-perspective view. We'll talk to her, grill her. She'll come out either crying or laughing. And then about 3:30, we'll see, we'll all convene at the Surfside over here to discuss, to celebrate, etc. With this, I'm going to turn the stage over to Catherine Furby and the presentation of her final PhD thesis.
Cool. Thanks for that introduction, Stuart. Thanks for coming. So today I'm going to talk about how the coral animal inhabits its own skeleton and all of the tiny organisms that live there with it, and then how that community structure enables the coral to recover from stress. But first, a short PSA for science, which I realize is preaching to the choir with this audience, but like, let's just take a second to remember why we all got into it. So science is about asking questions and taking risks, and I want to thank my committee for allowing me to really like run with that. I want to thank you guys for giving me the freedom to ask questions, but also the support for this long journey. So that said, a couple of years into my dissertation, I got this idea that maybe corals could come back from the dead. And we called it the zombie coral hypothesis. And it was basically the science equivalent of like a search for El Dorado, like an ancient ruined city and we don't know where it is. And so this presentation is going to be a story about that search and what we found along the way. And then before I get into coral recovery and the science I did, I just want to tell you how I got interested in coral recovery. So before I was a grad student at Scripps, I was a research assistant at the Woods Hole Oceanographic Institution in Massachusetts. And this is not a picture from Massachusetts. This is one of our field sites in the Red Sea. And the project that I was primarily working on at the time was looking at coral disease. And this, I think it was on my very first dive as a scientist ever. I came up from the dive, and we had done this transect on coral disease, and it came up, and my dive buddy was like, that was an amazing dive. Like, that was so cool. And I was like, yeah, it's awesome. It was really great. He was like, the corals were super diseased. It's amazing. And I decided at that moment, like, I didn't want to have a job where when the corals were sick, we were happy. I wanted to have a job where like we were happy when the corals were happy. So this is probably after that. So I wanted to focus on the positive parts of coral growth. And now we even have a hashtag for it. So Nancy Knowlton wrote an op-ed in the New York Times a little while back about how important optimism is in conservation. So with that theme, I'll start now talking about my dissertation. Okay. Okay. So I'm not going to use a laser pointer. Sorry. I'm going to use a regular pointer because I feel like it's more fun to like get up here and show you what I did. Okay. So we're going to talk about what it means to be a colony. And we're going to use a terrestrial system that I think we're all maybe more comfortable with, or maybe this audience is more comfortable underwater. But so this is a famous aspen grove. It's a cartoon of it. It's in southern Utah, and it's a colonial tree system. And what that means is that the root system is all interconnected. So these trees are all able to share resources. But additionally, you might think of a tree when it wants to reproduce, creating seeds, right? It makes a seed, it gets dispersed in the wind, it lands somewhere, grows a new little sapling, and that makes a new tree. But this tree has been able to actually short circuit that dispersal system. It can just take the roots and use it to grow a new tree. And so in that way, this sort of colony is able to then reproduce clonally. So that's kind of that system. And then if you take that one step further, you can think about if we lost one of these trees, this system it's not really bothered by the loss of that one tree, right? So this tree is all one genetic individual. And so thinking about that modular construction, we can take that and we can take that and talk now about corals. And I like to sort of do this at the beginning because we're unitary organisms, so being modular isn't intuitive necessarily. So with the coral system, in theory, all parts are created equal. And with humans, there are certain parts we can't live without. So this coral is able to lose pieces and then recover from that. Okay, and then just in case we need it, real quick refresher on what is a coral anyway. So here's a coral reef, looks familiar. The coral is what builds all those structures. Here's the colonial part, you can see all the polyps there, they live together, they share the space, and that forms this thin tissue layer on top of the skeleton. And if we zoom in even further, we can see that there's a microscopic algae called zooxanthellae, and that lives inside the coral with a symbiotic relationship. So this algae helps the coral get energy from the sun. And when I talk about a bleaching event, I'm talking about the loss of these algae. Okay, so how do we promote recovery on the reef? I promised you an optimistic story. We deliver it. Thinking about how corals recover, I'm going to go through different ways that corals can live and die on the reef. So here's our coral, sort of in outer space. And corals have been around for over 200 million years, so they've been through a lot. But recently, they're going through more than normal. So let's say this coral experiences a warm water event, it bleaches, and it dies. The first thing that's going to happen to it is it's going to have a thin carpet of turf algae start to grow on the dead part of the skeleton. Don't worry, this coral lives on a reef where it has fish that eat the algae. So the fish actually keep that algal growth minimized. Basically, these fish are mowing the lawn and keeping everything nice and low. And so that means that the coral is then able to regrow out over the part of itself that was dead. And maybe even if it gets really happy, create coral larvae that can then grow and seed new reefs and make new coral growth. Okay, but now let's say that our coral is not so lucky. It's on a reef where humans have eaten the herbivorous fish that eat the algae. And in addition, let's say we have put a sewage outfall plant near this reef. So now what we're essentially doing is dumping fertilizer into the water, and that is going to help the algae grow. And this can actually make it so that the algae is able to outcompete the coral in its own skeleton. And it can eventually lead to the complete mortality of the colony. And while algae are really cool and they're extremely important to this ecosystem, they're not reef builders. So this can lead to the eventual decline and crumbling of the reef structures themselves. And this is also an example of the algal succession, right? So it's the way that we can move through the coral death and the different communities that occur the longer the coral is dead. So sort of two different ways that we can think about how coral recovery might or might not happen on the reef. And as promised, I'm going to be focusing on this part here. So we're going to talk about coral regrowth. So how this coral is actually able to resheet over its own skeleton. And so when I say resheeting, I'm talking about it laying down tissue out over the skeleton. So if we were like to take a close look, this is the inside of the skeleton here. It's like a solid rock, and then the tissue on top is able to grow back out over it. Okay, so I'm not the first person to think about this idea. So other studies that we thought about early on that were inspiring. For this short note, that's looking at how the corals recovered from this bleaching event. So this is a thicket of coral. They've grown really, really densely together, and the surface has bleached and died. And what the study found was that there were cryptic tissues deep inside the thicket of this coral. And what they're talking about was this little fragment of tissue was actually able to grow back out over the existing skeleton. That's cool. So thinking about this, now we have to do like a little vocab. This is the only words you'll have to learn today, but there are two different ways that a coral can grow a skeleton. One is imperforate, which is what it sounds like. You can see there isn't a lot of space or holes in that photo. So that's a scanning electron microscopy photo of the inside of a coral. But there's another way that the coral can grow. Oh wait, yeah. So that's what it looks like. This is a very scientific illustration of what it looks like. But there's another way that it can grow, which is that it can have a perforate skeleton. So what that means is that there are spaces deep within the coral, but also those spaces can be lined with living tissue. So we're going to spend the rest of the talk exploring this part here, which is how this internal structure might aid the coral's recovery. And then one story that I feel like this is it. This is the moment where we started to really think about this. But so this is a story from the Keppel Islands. And what happened is the corals bleached, and then they were overgrown by macroalgae, just like in the cartoon example I gave you. But this coral was actually able to recover really quickly. So when the scientists went back to the spot and found that the coral looked like it had made a full recovery, here it is getting overgrown with macroalgae and then coming back. It happened too fast. It happened too fast for coral larvae to have dispersed through the water, landed somewhere, and grown a new coral. So they decided they wanted to take a closer look at what was happening. So they took the branches and they thin sliced them and looked at them under an X-ray. And they found that the coral had actually stayed alive in the base of the branches and then was able to regrow out over the skeleton. And this was really cool because we didn't really even know that the coral had the ability to do this. So it's laying down new calcium carbonate skeleton over the algae. And that's where that study ends. But now that I've told you how the different structures of the skeleton are, if you look at this picture, we can actually see that there are holes in this skeleton, right? And it's an acropora coral, so I can tell you it's a perforate coral. So it's possible that the internal tissues of this coral are actually able to help it grow out even faster. And there was another study done shortly after this in an aquarium that tested this exact fact. So it took coral fragments in aquaria and they scraped the tissue and a little bit of skeleton off and made an injury point in the coral, and then they watched the coral recover. And what they found was the coral didn't resheet. It didn't grow back over the top like you would expect, even maybe like when you get a cut and your scab starts to like resheet. What it did was it actually rose up from internal tissues inside the coral. So that looks kind of like this, right? So instead of resheeting over the surface, the internal tissues actually grows up and helps the coral recover. So that was super exciting to read about. So now I want to know, like, this is a cool idea. How far can we take this idea? At which point, I think maybe Forrest and I started talking about a different animal altogether. This is the Hydra. It's a cnidarian, so it's related to a coral, but you can think of it like a freshwater jellyfish. The Hydra is pretty famous in science literature for its regenerative abilities. So the head, I think, looks like a little bit like Medusa, so you can cut the head off and actually will grow a new head. And you can get real mad science-y, and they did, of course, and they were able to actually disrupt the entire organism. And they found that the cells of the Hydra were able to regenerate despite being completely in pieces. And that was really cool. So I wanted to know if the Hydra has this ability to be modular down to almost a cellular level, and we know that the coral can regenerate from these internal tissues, is it possible to look for that kind of modularity with the coral system? So this brings us to sort of like the central question in my thesis, which is, what are the survival advantages of a perforate colonial coral? And of course, we're going to do this, at least at the beginning, swimming around on the reef. So this is a pretty typical thing to see when you're swimming around on the reef. This is a big table coral, so that the coral is like this, and then this part is alive, and this part is dead. So this part has that filamentous turf algal mat that I described earlier, and it's pretty well mowed, so we know we're on a reef that has enough fish to eat it. Now I want to take a closer look at what's happening here. What's happening where the algae and the coral they're sharing a skeleton at this point, right? So here's a cartoon, and this is to show you all the different players that we're now going to go through. So we have the living coral polyps at the surface, right? This is the animal, and then deep within the skeleton we have this living tissue lining these canals, this pore space in the coral skeleton, living next to turf algae, which then also has endolithic algae and fungi deep in the coral colony. We even have some here sharing a space with the living coral. So I want to ask that question about how a coral is surviving in this space. and I want to sort of search for life within this system. So, do that. My thesis. There are 4 chapters. I have a confession to make, which is I sneakily already told you about chapter 1. So chapter 1 was a literature analysis of coral regrowth and recovery, which we just did. So now moving on to the 3 data chapters of my thesis. Chapter 2 just came out. You can read it online now. Photographic surveys of colony death and survival on the reef. So this is looking at the surface of what's happening with the coral through time. And then chapter 3 is internal visual snapshots of life inside the cellular matrix. So this will allow us to get the surface, but also what's happening internally. And then chapter 4 will go even deeper still, using microscopy and genomics to examine the micro-community structure within the coral. And this will go a step further in that we won't just look in the canals, in this pore space, we'll actually be able to get into the opaque, solid parts of coral as well. All right, so how are we going to do? We have 2 characters that we're going to look at. The first is Porites superfusa. It's a small encrusting coral that's localized to the Central Pacific. And then Acropocytheria, which is basically the opposite of superfusa. It's a big table coral. And we are going to, all of my research is based, supported by fieldwork at Palmyra Atoll, which is this remote island in the Central Pacific. It's about 1,000 miles south of Hawaii, so here's Hawaii. So we're in the middle of nowhere. It's part of the Line Islands. It's a US territory, and it used to be an old World War II military base. And I think Palmyra is a cool place to do research because it has limited local stressors. So Palmyra doesn't have to deal with a lot of pollution or fishing, and it doesn't even have permanent human habitation. So there's just a series of rotating scientists and visitors that are on the island. Okay, part 1, visual observations, starting with chapter 2, Porites superfusa mortality and recovery from a bleaching event. And the collection of data for this actually started before I even became a grad student. So Jennifer Smith's lab has been collecting photoquadrats at Palmyra, and when I came into the lab, we were talking about what had happened with the photopods, and we looked and we saw that in 2009, here's a picture that the Smith lab collected. But in 2010, it's kind of like an obvious coral missing. All of a sudden, superfusa disappears. So we wanted to figure out what was happening with the coral and how it was coming back or not from the stress. And this is also one of the most abundant corals on Palmyra. To do this, we're going to use the photopods, which I kind of just mentioned, but Maggie Johnson is here modeling the photopod for us. And the way this works is you put a couple pins in on the reef so that you can go back to the exact same spot every year or twice a year. And you can't see the pins in this picture, and actually you can't see them a lot of times when you're on the reef. But it's a great way to mark where you are, and you can do this along a transect, and you can also then do this at sites around the atoll. So it gives you an idea of how corals are changing through time all around the island. So I'm just going to walk through an example of what this looked like for us. So in this picture, I've actually traced every single superfusa that fits within the frame of the photopod. You can watch through time, like something terrible has happened, they're all gone, so they've bleached and died and even been overgrown by other things. And in red is highlighted where they're currently alive in 2010. In 2011, you see a similar story. So there's more death, but there's also some growth. So now you see they're like coming out from under things, they're starting to grow a little bit. Here's what we saw overall. So we saw a 75% decrease in superfusa cover. So this graph is looking at, this is through time, and then all the different lines, those are just different sites around Palmyra. And the sites are set up all around the island, so we get sort of a general idea of what's happening on the Furby island. And you can see that while there's a big decline, the post-disturbance trajectory, it's a little uncertain still. There's some growth, but there's some death. Okay, so then to break that down, where I wanted to know what the recovery mechanisms were. So where's the growth coming from? Where's the death coming from? And so to do that, we'll just do a quick little, so if you're a coral, you can recruit, right? You can be a coral larvae, you can come in. You can also completely die. You have some other options. You can clonally grow, or you can partially die. And so the thickness of these arrows are actually, they're based on the average of our total theta. So you can see that there's more death than growth overall, right? But we also see what's interesting is that complete mortality and partial mortality are equal. They're equally contributing to this decline. And then on the growth side, a surprise to find that clonal growth was as important as recruitment, which I'm pretty sure before this chapter started, I had all my horses on. I was betting on the recruitment to be the answer, but it wasn't, right? It was both. And then in addition to this, we created another category of growth, which was resurrected recruits. So what this is the coral was alive, the next year it was dead, and the next year it was alive again in the same place where it had been previously dead. So there are a few things that could have happened. It could be that this is just a really awesome place to be superfisa. So the new coral came in and landed there. Or it could be that the coral tissue survived in a crack, in a crevice, or maybe it was too small for the photograph to pick up. And that category accounted for 12% of the growth that we saw overall. So you can imagine why at this point in my thesis, I'm thinking corals maybe come back from the dead. That wasn't the only thing we found. So this is a super rich data set. You can get all kinds of demographic information on the coral this way. One thing that we found was that survivorship was size dependent. So this graph is showing us colony size, but then it's a histogram, so it's also showing us the frequency. So white is it died before 2012, and black, the black bars are that it survived through the time point. And what this graph is showing you is a few things. One of them is if you were a big coral colony, you survived the entire time. And this kind of makes sense, right? So let's say you're 16 centimeters of coral, and something comes through that's going to kill 8 centimeters. That's okay. You still have 8 centimeters left. You can work with that. But if you're an 8 centimeter coral and something comes through, that's it. You're gone. Like you can't come back from that, right? And we saw that reflected in the data. So we saw that mean colony size declined over time, and we also saw that the number of colonies declined. Okay, so this chapter is looking at regrowth, right? And we found that regrowth and recruitment were equally important recovery mechanisms. We've coined a new term, resurrected recruits, and we found that survivorship is size dependent. And the question that we were asking was, how does this colonial mechanism impact the coral's response to stress? What we found was that partial mortality significantly contributed to coral reduction, and regrowth was an important recovery mechanism. And I think that these mechanisms can help inform our understanding of colonial corals in general. But obviously I'm left with this like burning question after the study, which is, did the cryptic tissue survival play a more important role? Where is this coming from? I really want to know. And so to do that, I'm going to need to do something more than just take a picture of the surface. We're going to have to go like deep inside the coral skeleton. And so we're going to get super tiny. And before we get super tiny, we're going to do it in a system that's really big. So the rest of the thesis is going to be talking about Acropora tables, which it's hard to pick a favorite coral, you know, but these guys are great. So these table corals, but we average sampled them at about 12 feet in diameter, but they can get enormous. So Amanda Carter is 6 feet tall minus her fins. So that gives you an idea of how massive this coral colony is. And I cannot even describe what it's like to swim on a coral that makes you feel tiny. Like if there was ever something I wanted to share to encourage conservation, I think it's being in an ecosystem, feeling small. So when I first started doing this project in Palmyra, I was swimming around these big corals, just like, these corals are amazing, they're so big, they must be ancient, like what have they seen? And as it turns out, Acropora grow pretty fast, so they're not super old. They're actually way more dynamic than I ever would have expected them to be. So for example, here's a photo of one in 2011. Gareth has tagged a bunch of these. So this is like a perfect looking table. It has these little folios, spirals, it's symmetrical. In 2016, it's looking like this, right? It's a kind of a broken mess, but there's still living coral here, right? This is the live part, upwards, that spiral still going strong. These things are growing and changing in all kinds of ways. They're able to regrow over their own skeleton. They're even able to, sometimes we found them, they were popped off the reef, like flung down, flipped over, and then they're growing back up from the underside. This dynamic system, it's an amazing system to study. And one of the things that our lab does is create these 3D reconstructions of corals. So they were kind enough to do this for one of my proper tables. All right, so how are we going to sample these massive things to look tiny? For the rest of the chapters, we're going to be looking at coral cores. And so here I'm going to show how we sampled it. The first thing you do is you take a bunch of Home Depot parts that are not supposed to go underwater, and you attach them to a scuba tank, and you take them diving. And then you're able to use this pneumatic drill and the hole saw to get into the skeleton. And then just to give you an idea of what that really feels like, because I think when I describe it, it doesn't really capture what's happening. I'm using a power tool underwater. But when you're done, you get this neat little hole in the coral. You pop the core out, you refill it with epoxy. The coral is then able to re-sheet over the top, and you have a sample that you can now spend the rest of your life looking at. And that brings us to chapter 3, exploring the possibility of an intraskeletal circulatory system in a perforate table coral, proper synchronia. And I did this work with Nick. Now we're going to take the cores into the lab and we're going to thin slice them. This looks like so easy, but it's actually pretty difficult because you're wanting to thin slice a piece of rock, basically, but you need to do it gently because if you disrupt the system, you don't get to see the structure of what's living in it. So we do that with the coral colonies. This is like a table saw, right? So we're thin slicing it. And when we're done, what we get is basically like a slice of coral bread. So now I've taken this coral core and I've sliced it up really neatly. And the reason that I want to do that, right, is because I'm trying to get at a cross-section of what the community looks like internally. Okay, so here we go. So here's a slice of coral bread. You can see here is at the surface, there are coral polyps. Maybe it looks a little bit pink. And then this way, we're going now deeper internally into the coral skeleton itself. And then if you look at a dead coral, we see that the surface looks like something that rolled out from under your bed. But also that internally, right, the skeleton, it looks familiar. And Greg Rouse was kind enough to take these microscopic pictures for us. So that's great. So the first thing that we're going to do is, all right, my question is, I want to know what's happening inside the skeleton. So we're going to need to get rid of the skeleton. So we put the samples in a mild acid. We're actually able to gently dissolve away the skeleton itself. And what you're left with are just the squishy bits. And this is the tissue. This is how the tissue is lining these intricate canals inside of the coral skeleton. And then we're a little surprised to see that at a certain point it just becomes this weird fuzz. I think it was kind of a frustrating long time that we just called this the fuzz. So I'm going to just call it the fuzz for now. But when we dissolved the dead coral, we saw something similar. Except now we're really just in the weeds, right? At the surface, you can see there's some algal structures here. But once we get deeper down, it just becomes this massive lint ball. So we're going to need a different microscope. We're going to get help from Malcolm Wood, and we're going to use what's called a scanning electron microscope. This is a really fun toy if you still need a chapter of your thesis. I highly recommend it. You take the coral piece, you're able to mount it with the silver paint Then you put it in this instrument that sprays iridium on it, and it looks purple. And what you're able to do eventually is to create a 3D construction of your coral using electrons. And it's a little bit like, if you remember Miss Frizzle from the Magic School Bus. So it's basically what we're going to do now is shrink down really small and walk around inside of the coral. And so here's the first thing that we see. So we look at the live coral near the surface and we see, so it kind of cracks this open now, so it's not a thin cross-section like you've seen before. But you can see here, this is the tissue lining this hole. And it looks like in the process of ripping it apart, the tissue's not well attached because it actually looks like it's starting to peel off here. But you can see here where it is still attached to the skeleton. And if we zoom in, we get this. So this is taking a closer look at the internal canals of a living coral. And we see that it's flagellated gastrovascular canals, which means that this coral has all these little fine hairs. And just for scale, just for the rest of this talk in the corner here, I have, this is the width of a human hair. Just to give you an idea of how small we've gotten at this point. Okay, so if we zoom into these canals, you can see really, really well what they look like. And maybe you think of a coral as it's like a passive, still thing. But these corals are actually able to move all kinds of stuff internally. So these hairs are able to move. They have fluid inside of these canals that they can actively move back and forth. In addition, they're able to move their zooxan belly around in their tissues, depending on the light. And then this is our human hair again. So we're super tiny. Like the screen is the width of a hair. Okay, so we found that near the surface of the coral colony, there are these tissue-lined canals. But we saw that once we got just a few millimeters in, that it becomes bare calcium carbonate with these fine tendrils of something in there. So that's our fuzz, right? There's not much else in the interior of the skeleton other than these. And so we're actually able to separate it from the rest of the mass and get a good look at it to see that it's actually an endolithic fungus that's living underneath the live coral tissue. Just to give you an idea of how small they are now, there's the width of a hair. Okay, so now let's do that with the dead coral. So we have this dead coral, we're going to take a look at it under the SEM, and we see that the surface of the dead coral looks like a Tim Burton creepy forest. And then all of this pore space is just filled with stuff. And you can actually see it here too, right? It looks like it's filled with sediment or something. What we see is it's filled with endoliths. It's filled with algae, it's filled with fungi. It's this crazy map in all of the pores. And they're really, really tiny filaments at this point. And in addition to filling all the available pore space, we also see that there are microbores that have drilled their own holes through the skeleton. Which is why when we decalcified it at the beginning, it just looked like this mass. Because the skeleton is being basically Swiss cheesed by all these microbores. It's completely packed with stuff. We can take a closer look at who's there to a certain extent with this microscope. So we can see that there are fungal fruiting bodies, some mysterious algae. There's Ostriobium, which is a really well-known endolithic algae in corals. And then, just for fun, because we can, we're gonna go like really tiny at that spot right there. And this, the bacterium that's growing onto the surface of an endolithic algae that's like inside of the coral. So now we can see really, I can't even show you the human hair at this point. But thinking more about how the coral is functioning in this space, it's not a passive neighbor to all these organisms. So the coral is actively defending its territory. And this is a bit of a mystery to us, right? So this is a cascade of calcium carbonate granules that look like they're pouring out of this hole. And it could be a couple things. It could be that there's a little microinvertebrate inside of that hole, like kicking up sand as it digs through. But it also could be that this is actually the coral itself had done this. So there's a paper that has shown that coral that lives on top of endolithic algae or endolithic fungi actually has the ability to try to defend itself. So they did a study where they found that the coral was creating calcium carbonate granules and spitting them out down at the fungi. So the idea is that it's maybe trying to create some space in between itself and this organism that's living directly underneath it. There's one last thing that we found in this chapter that was surprising, and we found it in both the living and the dead coral. And the answer is nothing. So deep within both treatments of coral, we found these deep, empty, open skeletal canals. And there are a few things in here. So there's this few strands of fungi that we found inside, but for the most part, it's surprisingly barren. Actually, I think Nick said there's not as much biology as I thought. That's how I thought. We get down here and it's empty. But that's still interesting to think about, right? Because although maybe there aren't a lot of organisms in there, it must be filled with something. So this is filled with seawater, or maybe it's filled with some kind of fluid. If the coral is actively pushing fluid around in the shallow space, is any of that fluid being passively moved around? You can imagine that it's an interesting idea to think of a living and a dead coral in one block. So I think normally when we talk about corals, we think like this one's alive and this one's dead. But in this case, and very commonly on the reef, they're sharing a skeleton. So they must be interacting with each other, right? It's constantly changing dynamic. All right, so to wrap up, living and dead coral host a dense community of endoliths. The deep skeletal canals in the peripheral coral skeleton, they have less biology than the shallow surfaces. So my question was, what is the environment like living inside of living and dead coral? And are there signs of life that we can discover, these living coral stem cells? So going back to the Hydra idea, we're wondering if we would see any sign of coral life in the dead part, right? But we didn't. So there's a limit to life in the coral. And we know that the organisms are limited. They're limited by light. They're limited by nutrients. These empty pore spaces are, I think, an exciting place to think about researching more. They're filled with fluid, but they might affect coral composition and recovery. And then the answer is that the dead skeleton is crowded real estate. Like those endoliths are fighting for space. And I don't think that it would be a good place for a living coral cell to wander in the first place. I think it's unlikely that that's our silver bullet answer. Okay, now we're gonna get really small. The last chapter was the hardest, but also the most fun. So I did this chapter, Beverly French, and it was difficult because we didn't have really a playbook. No one had asked this question before, so we were struggling a little. But the answer that we find is a list of community members in living and dead coral. So there was one day where I spent like a solid 8-hour day just reading about tiny things that live in coral on the internet to try to figure out what our data even meant. And at that point, I'm texting Beverly like, look, I found this cool shrimp. Kind of looks like a lorax. You've just been staring at images forever, trying to make sense of them. This is an ostracod that we found in one of our samples. Okay, so that brings us to chapter 4, corals and their neighbors in partial mortality. So we did multi-marker barcoding and microecological exploration of living and dead coral tables. And the genomics part of this work was done in Eric Allen's lab with Beverly French and Jess Blanton. And the microscopy part was done in Jennifer Smith's lab with Emily Kelly and Sam. All right, so we're back at this big acropora table. Only now in this photo we zoomed in, so this coral is actually taking up the entire frame. This is just like a sort of a top tier of the table. So this is connected by living tissue to this plate. It's just a multi-plate system. But what I want you to notice here is that there are dead patches on the coral, right? So this coral is resheeting, regrowing. This is a big dead patch here where that ruler is. So if we go even closer, we can see how we took the samples. So now instead of just living in dead corals, we're also thinking about living corals, but then 2 treatments of dead corals. close to the living tissue and dead corals far. And the reason that we sampled like this was because I wanted to ask the question of how is this community changing as the coral dies? So I made an assumption at the beginning that a dead coral close to the living tissue would be more recently dead. And like, spoiler alert, I can tell you that the data supported that. So I think that we did sample in the correct way. Okay, so how do we do this? We did what's called meta barcoding. We used a few different primers, but basically what we're doing is we're using genes to search for different types of organisms. So the reason that we use multiple markers is because some of these are, they're better than others for looking at different kinds of organisms. And I wanted to get the widest variety view of what we were seeing. You'll be happy to know that live corals are different from dead corals. That was our first result. Pretty groundbreaking stuff. But now we get a more detailed view of what's there. So we did find a cropper actually in about half the samples, but what our main marker for living coral was actually the zooxanthellae. So the live coral had a really strong signal of the symbiotic algae loads in the coral. And it also had a small amount of other things. So a heteropont you can think of as kind of like a slime mold. And then these are sort of the main additional organisms that we found in the system. So we have sponges and red algae, green algae, and fungi. Right, so moving to the dead corals that were close to the living tissue, we find what I described in the original cartoon. We find this fine filamentous carpet of turf algae. And we can take a closer look. This is Gelidia, Caudifera. These are these fine filamentous algae that are growing here. And we get a good view of a few different things, but it's not a ton of information. And this just for fun, you guys, is crustoscoralin algae, which when you're on the reef, it looks like this. It's pink. It's this pink cement that grows all over the reef, and it's known to promote coral growth. So this is a good marker to see on the reef. Okay, so with the genomics now, we're going to broaden our perspective of what we're seeing. So here's the microscopy stuff, and this is just the major players that we found with the barcoding. So there's a lot of red algae. Here's our crustoscoralin algae again, and then we see some other things. There's sponges. There's this like weird peanut worm. And then if we take everything, now it looks like this. So with this method, we're able to really expand what we see. We also saw that the microscopy, what we saw under the microscope matched what we saw with the genomics. Okay, so moving on to this second treatment of dead coral. So now we're far from living tissue. You can see that there's a lot more of the pink crustoscoralin algae. We have some familiar faces, so the Gelidia is also still here. And then if we add in the genomics, it gets even more complicated. So now we found a way stronger signal of the crustoscoralin algae, and that actually goes along with how we think of reef succession on Palmyra. So I think one of the reasons that Palmyra is a really interesting place, in addition to it having these limited local stressors, the crustoscoralin algae on Palmyra is just bonkers. So we know that after the coral's dead for a certain amount of time, we see this pink cementing, calcifying algae come in right away. And that, I think, may be one of the keys to Palmyra's recovery success. Okay, we also see other things. We see crustaceans. Polychaetes are like these little sort of scary-looking worms up close. And then if we add in all of even the tiny things we saw, we see that this community is diverse, and it's also very different than that previous dead coral. So we put it all together, we see that there's a systematic transition of succession on Palmyra. We can watch the community develop over time. And the first thing that's really obvious, right, is the live coral is way lower in diversity than the dead coral. And that makes sense because the coral is actively defending its space. It's making it harder for things to land on it and also for things to bore underneath it. And then the longer the coral is gone, the more we see the community change as things come in and bore holes and make a dynamic community. Okay, so after all of that, For the 18S work, we found 43 out of 320 OTUs, which basically means that we've taken this information and we've compared it to a database, and we have a lot of unknowns, which is classic, right? The more you know you don't know. But this could have happened for a couple of different reasons. It could be that we have strange hybridizing organisms in here, or it could be that a lot of the things that we sequenced just haven't been sequenced before. We're going into a cryptic environment that's pretty difficult to study, so I think it's reasonable to assume that we might be discovering something strange and new. But going back to this idea of thinking about living and dead corals sharing a space, I want to talk about the players that overlap. So these are the things that we found in both the live and the dead coral samples. So if we want to ask questions about how living and dead coral are interacting with each other, I think it's interesting to think about the community members that they share. So we have red algae and green algae, fungi, sponges, and protists. And the first question I wanted to ask when we were thinking about this was, okay, is it possible for the endoliths to start shunting resources back and forth? Like what is the extent that these endoliths have the power to change what's happening in the skeleton? And so to answer that question, you can really only look at organisms that are cenocytic, so organisms that have this ability to move resources around, essentially in a tube. And so in that case, we're only going to be able to look at two organisms. The first one is Ostriobium that we saw from the SEM images in the previous chapter, and the second is an endolithic fungi that we found in almost all of the coral samples. And so I think for a lot of this talk, I've been talking about how corals are competing with algae and how there's this negative interaction between them, but it's not all terrible. So some endoliths do have the ability to work positively with the coral. So there's one study that actually showed that during the bleaching event, the coral have lost their internal zooxanthellae, but because of that, there was actually a bloom of the endolithic algae underneath the coral. And the algae shared resources with the coral, and that coral that had the algal bloom underneath it survived the bleaching event longer. And then for the fungi, there was a study done that they put fungal and algal cell, or fungal and coral cells together in culture, and they found that the fungal cells promoted growth of the coral cells, specifically the coral cells that create the skeleton. So it's kind of interesting to think about because you know that the fungus is living underneath the coral. So maybe it's wanting to encourage skeletal growth because that is the home where the fungi lives. But that's, who knows why it's there. But so this is just two examples of how the endoliths can be good in addition to being bad. Okay, so wrapping up, we've taken microscopy investigations and metabarcoding. We're able to correlate what's living at the surface of the dead corals. Session using genomics. And we found a large community of endoliths and cryptic invertebrates that are living inside the skeleton. And we've learned enough that now we know we have a lot more work to do. So the question we asked was, how do the cryptic communities change from living to recently dead coral to far dead coral inside the skeleton? What we found was there was a significant difference in diversity between the living and the dead. We also marked changes in the surface community along this algal gradient, specific, I think, to this Palmyra system. But I think the main takeaway from this is a cryptic environment. We're talking about inside of not just the pore space of the skeleton, but this opaque rock that the skeleton is building. And so this is the best way for us to get a glimpse into the micro-community structure. I think the changing tiny communities have the ability to affect the host coral's survival. Okay. All right, so going back to this original diagram. So we decided I decided, I guess, I was gonna search for life within this system. So we found that perforate corals use regrowth as an important recovery mechanism. We looked at the surface and Parietes superfusa. As we go deeper into the skeleton, we see that endoliths are filling all available spaces in this narrow range where we found life. And then there's a limit to the life inside the perforated skeleton. So there's also an internal subway system connecting the living and the dead coral. We've expanded our idea of the coral community. It's way more complex than you can see with the previous microscopy explorations. And these tiny dynamics may control coral recovery from partial mortality, which we're seeing more and more of. All right, so dead corals are alive in a way, right? They're filled with things. But zombie corals are still dead. So we did not find free-range coral cells inside of dead coral skeleton. I think it's very unlikely that they can live there. So this silver bullet, save the world answer doesn't exist. The answer is more nuanced. These tiny communities are complex and they're affecting the coral's growth. But there are like also some things we could think about if we still wanted to explore the zombie coral idea. So these deep perforate skeleton, it's connecting the living and the dead. It's filled with some kind of fluid. It may be passively moving things around inside of the coral. And in addition, we have these endolithic organisms that are living in both live coral and dead coral in the same coral skeleton. So in conclusion, the colonial nature of coral growth, the inclusion of this perforate coral morphology, and the endolithic community present in living and also adjacent, attached dead coral skeleton, these things are important for colony survival. They affect how the coral grows in its environment and whether it can regrow once it's been damaged. Okay, so as we're getting into summer 2017, I just want to say, like, my perspective on what's happened as I've been a grad student for the last 6 years. So when I first came into the lab, we were working in Palmyra, our lab was working in Palmyra, and we were talking a lot about how limiting local stressors was enough. And I used to tell people, if we can just leave corals alone, like they're gonna be okay. We can just reduce pollution and fishing. The corals have an amazing toolkit for survival. I think now it's getting more complicated, right? Like we're seeing more and more warm water events. They're becoming more common. But places like Palmyra are still surviving. Like they're still doing well. So I think that research on how corals recover can help us understand how to set them up for success. And then I'll end my science PSA by saying that science is not just about hypothesis. It's also about exploring. Okay, so with that, I want to thank my funding sources. I want to thank Jim and Marianne Beister for coming, for supporting me in my last year of my PhD. Before that, I had the National Science Foundation helping me out, and I'd like to thank CNBC for constantly supporting me with science, but also with my communication ideas. I want to thank everyone in the grad office. I'd like to thank my committee. I've been calling you guys super friends behind your back. When you get like Batman and Superman and Wonder Woman together, you can solve all the problems. Thank you guys for supporting me through all this. Stuart has been one of the best advisors you can ask for. He's a great person to go talk to about big ideas. He's also there when the big idea doesn't work and you need him to talk. Okay, I want to thank Elsa for always having really solid, amazing advice, and I feel like basically teaching me how to write science. Forrest, I'm gonna blame you for the zombie coral. I don't remember what we were doing when we thought of that, but it was yeah. Lisa always brings an amazing perspective to the committee meeting, and I'm so happy that I was able to include you in this process, even if I had to watch you and Forrest argue about aesthetics. And Jen has been my advisor when I'm just my second advisor, basically. Our labs have worked so closely together, and I've really appreciated your technical and advice and just general advice. Okay, there are two people that basically saved zombie corals, and the first one is Nick Collins. Your support through all this, understanding how field biology works and how the lab works, has been like incredibly important to me. Oh no, do you get feelings? Okay, Beverly, like, you guys, Beverly is the most amazing person to work with. Like, she's the hardest working, friendliest, like, most fun person to be with in the lab at like midnight. Thank you so much for your help in this last chapter. Okay, all right, so I did a lot of field work for this. I did a lot of lab work, but I also did a lot of field work. And I want to thank the staff on Palmyra and also the staff here at Scripps for your guys' support with all this stuff. It's good to feel safe when you're doing crazy adventure things. I want to thank the communications office and the Birds Aquarium and Eric Allen's lab and SCSU Writing Club, RIP, and Dimitri's lab for all of your help with the fluorescence work. My lab! You guys are great on a boat. Yeah. I really appreciate everyone's support, putting up with me and in the field and at home. I love you guys. And then thanks to my friends. Grad school's pretty fun. Stay in school, kids. And then I want to thank my family for coming out here from all different places and like supporting me remotely. I want to thank Noah, who's not here, but who's probably inside that computer. I'd like to dedicate this talk to Rachel Morrison and all of her friends who are still with us. And I would like to dedicate it to everyone who believes in something. They still have hope. Thank you.
So we have an opportunity now for the public to ask questions of Kate. Hand her some softballs so we can hit her with hard balls later.
Yeah.
So at the beginning, you showed some Acropora that seemed to be dead, and yet they can regrow from the inside out. So how is that different from zombie corals that you said go with this?
Zombie corals were with us all along. So I had this idea that maybe that internal tissue could survive disconnected from the living tissue. So I wanted to know if like a few of those cells or maybe a small patch of that tissue inside the canal could actually survive inside of that dead part underneath the turf algae. Because of the way that we saw them regrow in the first chapter with the superfusa, they were coming back in a place where like previously I'd seen CCA or turf algae or something. So I thought maybe they can survive like underneath that. So yeah, I guess they're there and they're. Thanks, that's a good question. Yeah.
This is less of a question and more of a request. Can we talk about reshooting once you come to your defense party?
Yeah. Mm-hmm. Yeah. Reshooting this. Yeah. Definitely.
Awesome.
Thanks for that question. Yeah. See here.
In your graphs, I don't know if I was reading the color coding wrong, but it looks like you're still seeing zooxanthellae signals all the way through even the far dead corals. Was I looking at it the wrong color?
The color in the pie graphs are a little like, it repeats a little bit of colors.
Okay.
We did see a faint signal of zooxanthellae in the near dead coral. I can't remember what the signal was, so I think it might have been too low for us to really count.
Okay.
But
Something else.
yeah, but that was something that I was wondering is if zooxanthellae could be in the dead coral as well. I think that's a cool idea. Yes.
follow-up question.
Yeah.
This one is an actual question.
Okay, cool.
How confident are you in the ability of the test to tell the difference between zooxanthellae and other nonflagellates?
Yeah, that's actually a really good question. So the way the zooxanthellae was coming out with the barcode of life, it's coming out pretty high like taxonomic level. But when we ran a known zooxanthellae sequence through that same barcode, it came out in the same place. So that's why I feel confident that it's zooxanthellae, but it could also include something else. Yeah.
So you didn't really find the zombie coral in the acropolis? Have you looked at other taxa?
No, I haven't. I really want to drill into the superfusa, actually. Yeah, I haven't. Next student can do that though. It's wide open. Yeah, it's possible.
So I'm fascinated with the little flagella that are inside of the coral. I'm curious how much we really know about them and whether they're maybe capable of injecting zooxanthellae when they get stressed. What's your thoughts about all that?
I don't know if the flagella can inject zooxanthellae, but I do know that they can. So there was this one paper that looked at something called the wandering coral cell, where they thought that the coral cells could actually like pick up and move around inside the skeleton. We didn't see any evidence of that. We looked really hard to see if we could see anything that looks like the coral cells are pretty obvious on the SEM, and we didn't see any evidence of something like that. Elizabeth Gladfelter, if you know her, she did some work where she actually sampled the fluid inside the coral and found out like chemically what was happening in there. So I think that's pretty cool. I'd like to do that with the dead in the dead space as well. Thanks, that was a cool question. Yes, monsieur.
There's been like you spend a lot of time describing what's underneath and how much you could influence, all in a positive way or negative, especially at the end of the talk. I wonder, is it possible to take a coral as alive and trim out that part, see how the coral adapts to this endolithic community? Is it here?
Like find a way to blast the endoliths out of the skeleton?
Yeah, I feel like
Yeah, I think they would go for it. I think if you remove the endoliths, that they would just like grow as fast as they could into that empty space. Yeah, that's just pure speculation.
All right, let's take one last opportunity to congratulate Kate. We're gonna follow this. Like I said, we'll be here for about an hour. We can meet you all over at Surfside at 3 o'clock. Those of you that don't know Surfside, find somebody outside, have them direct you there. But for right now, I want to congratulate Kate on a great talk.
Yeah, was that like a Australian mail series?